Be ready in a moment. Okay. So we can see the PowerPoint now?
Yes.
Okay, fantastic. Okay, cell structure. We're all made out of cells. Bacteria are single cells. Viruses, we're not exactly sure what they are, but they sure can cause problems. So I'll read through these text in windows. Cells have different sizes, different functions, different locations. We have smooth muscle cells in our digestive tract, nerve cells in our brain, and so on. Cytology is the study of cells using light microscopes and electron microscopes. Cells are surrounded by a membrane, which has extracellular fluid on the outside of the cell. There's intracellular fluid in the cell. The cell membrane controls what goes in and what comes out of the cell. So make sure you know all of these structures. Make sure you know what they are, in general, what they do. Okay, what's the plasma membrane? Separates the cell from the extracellular fluid. It provides support, regulates exchange with the environment, isolates the inside of the cell, and it responds to chemicals and other stimuli. So it's made up of a phospholipid bilayer. So what does that mean? So here we have the cell membrane. It has proteins that allow channels to form. These channels can be open and closed. The hydrophilic heads, the hydrophobic heads rather, the heads are hydrophilic, they love water. So all of these portions either are facing the extracellular fluid or facing the intracellular fluid or the cytoplasm. So you should know that phospholipids have a hydrophilic head and a hydrophobic tails, like dissolves like. So this is called a lipid bilayer. So make sure you know that carbohydrates are attached to the lipids. These carbohydrates are cell markers. These would be glycolipids. These would be glycoproteins. Why? Because this carbohydrate chain is attached to a lipid. This carbohydrate chain is attached to a protein, glycoprotein, glycolipid. These are very, very important for marking the cell with its signpost saying, I'm a liver cell, I'm a stem cell, I'm a blood cell, and so on. So make sure you know all of these structures. They'll be on WebAssign. Again, this is called a glycolipid. This is called a glycoprotein. Each one of these hexagons represents a sugar, a 6-carbon sugar. So what do these proteins do? They receive information. They allow substances to move through their channel from inside to outside or from outside to inside. They carry substances. They act as enzymes. The glycoproteins and the glycolipids act as identifiers. So make sure you read through these. Okay. Read through these, the function of proteins and make sure you know and understand these terms. So membranes allow transport. What moves through the cell membrane? It's selective. What goes in or out depends upon its size, its charge, its shape, and how soluble it is in lipids. We have passive transport, where substances diffuse from high to low concentration. Filtration can also uh, allow the cell to choose or allow the cell to determine what comes in. There's facilitated transport, which has a special channel to allow a substance to come in. Active transport has to have energy. So diffusion goes from high concentration to low concentration. Osmosis is the movement of water across a membrane from a higher osmotic pressure. Okay, so movement of water across the membrane goes from lower to higher osmotic pressure. We'll see what that means. So diffusion, so if a substance is dropped into water, it's going to start diffusion from high concentration till it reaches equilibrium. So diffusion depends upon the concentration of a substance in a liquid environment. So if you have this lipid bilayer, uh, some molecules that are lipid soluble can go in and out. Other large molecules or charged molecules depend on protein channels or protein pipes. So lipid soluble molecules like fatty acids are able to move in and out. Large molecules that can't go through the membrane have to be carried in through a channel or allowed to move through a channel. Small water soluble substances. So the whole purpose of this bilayer, lipid bilayer, and these channels, these protein channels, is to control what goes in, what goes out, and what goes in and out is depend upon the lipid solubility of a molecule, its size, its charge, and whether it's water soluble or lipid soluble. So what is osmosis? If this is a selectively permeable membrane, you can see over here that there's more H2O. The water molecules, the little blue dots are water. So osmosis moves from high water concentration to low water concentration. And that movement of water generates pressure. Osmosis is very important for movement of substances in and out of cells. We'll talk about it in the kidney and so on. So read closely through these diagrams. This is called a hyperosmotic because there's more dissolved substance. This is called hypoosmotic. Okay, so things tend to move toward equilibrium unless the cell or other chemicals in the body stop it. Okay, so here's the terms I use. We can also use hypertonic, hypotonic, isotonic. Isotonic means that the solution that cells are in have the same concentration as what's inside the cell. Hypertonic solutions have more dissolved material. Hypotonic solutions have more water. So we'll see what that has to do with red blood cells. So when you have fluid, IV fluid given to you in a medical emergency or a medical situation, the amount of substance that moves out is equal to the amount of substance such as water in and out of the cell. So the cell keeps its shape and is not destroyed. If you have a hypotonic solution outside of the cell where there's more water, that water is going to move inside the cell and cause it to burst. If there's a hypertonic solution inside the cell, meaning less water, or there's more water here, the water is going to move out of the cell. So let's start that again. If the cell has solution around it where there's more water, water is going to move from hypotonic to hypertonic. So the cell will swell and burst. You see in this solution, this cell in solution, there's more water in the cell than compared to the outside because there's a high concentration of solutes such as salt or water. So that substance, water will move out of the cell, causing the cell to shrink. Okay. Filtration is based upon the hydrostatic pressure of the water. So there's higher concentration of water. Water's going to cross the membrane. Solutes follows water. Filtration is important in the production of urine. Okay. Let's stop there. We'll have one video. Okay, stop recording. Stop recording. Stop recording.